Hello everyone, today we'll be talking about chromosomes. First we'll talk about prokaryotes. Prokaryotes have two types of DNA, chromosomes and plasmids. Plasmids are a genetic structure in a cell that can replicate independently of the chromosomes, and it's usually a small circular structure as shown on the top right picture. The main features of plasmids are that it contains naked DNA, which means that it's not associated with histone proteins. They are small circular rings of DNA. They are not responsible for normal life processes. They commonly contain survival characteristics such as antibiotic resistance. They can be passed between prokaryotes and they can be incorporated into the nucleoid chromosome. One important thing to note about these plasmids are that not all prokaryotes have plasmids, but they can contain plasmids. Genes are heritable factors that control or influence a specific characteristic about yourself. For example, the type of earwax that you have could be wet or dry and flaky. Depending on which gene you received, it could be one or the other. These different types of hair colors are also known as alleles, which are different forms of a specific gene. The gene in this case is the hair color, and the alleles are the individual different types of hair colors. Now we'll talk about eukaryotic DNA. Eukaryotic DNA is in the form of linear strands of DNA held in a helix. And notice that eukaryotic DNA is linear, while prokaryotic DNA is circular. Because eukaryotic DNA can be extremely long, if it's stretched out, they must be put into a suitable form to allow for processes such as replication and protein synthesis. And this form is called a nucleosome. This is formed by wrapping DNA around 8 histone proteins. Eukaryotes possess multiple chromosomes, and in humans there are 46 chromosomes. These chromosomes can vary by length, position of the centromere, and genes at their different locuses. A diploid nucleus has 2 of each chromosome, while a haploid nucleus has 1 of each chromosome. An egg and a sperm are considered haploid cells because each contain 23 chromosomes, which is half of the 46 in humans. When the egg and sperm combine to form a zygote, the amount of chromosomes within each combine together, and there are 46 in the zygote. So that means it's a diploid cell. On the picture it goes around, and when you reach to the point of adulthood where they're able to produce their own sex cells, the egg and the sperm, they go through a process called meiosis, which is called reduction division, which is where a diploid cell becomes a haploid cell. Homologous chromosomes are the same size and structure. They carry the same genes at the same loci, but remember that even though that they have the same genes, it doesn't mean that they're identical. The alleles can be different. The image on the top left is a karyogram. A karyogram is a depiction of how many chromosome pairs there are within an organism and also how the chromosomes look. Notice that each chromosome doesn't have to be the same shape as their homologous pair. And also notice that the very last pair of chromosomes is an X and a Y. Males contain both an X and a Y chromosome, while a female contains just an X and an X chromosome. The X chromosome is a lot bigger than the Y chromosome, but the Y chromosome contains the SRY gene. When this SRY gene is activated, the development of the male genitalia will occur. The last thing we'll talk about is about Down syndrome. Down syndrome is also known as trisomy 21, in which there are 3 chromosomes in the 21st pair. This usually occurs during non-disjunction during meiosis. As you can see on this graph, the chances of having a child with Down syndrome exponentially increases with age. However, from 20 to 29, the chances barely change. But from ages 30 and above, it starts to rise exponentially. We can determine if a child has Down syndrome before they are born through amniocentesis or chorionic villus sampling. Amniocentesis is a process where a needle is injected to gather amniotic fluid. Amniotic fluid contains cells from the fetus which allow for karyotyping the chromosomes within the fetus. So it allows us to see if there's trisomy 21 or any other sorts of possible diseases. Chorionic villus sampling is when a biopsy needle is inserted into the vagina to collect cells from the placenta. These cells contain genetic information from the fetus, which allow for a karyogram to be produced. Once the karyogram has been produced, you can look at the karyogram and at the 21st pair. If there are 3 chromosomes in that area, then the child has Down syndrome.